Hello everyone, this is Kellen Cavanero, and welcome to another episode of Inflammatory Content, the podcast about immunology and biomedical research more broadly. I'm here to interview world-class researchers to broadly communicate the cutting-edge work going on in their laboratories, and to get to know the people behind the science. So sit back, relax, and let's dive into some science. And if you enjoy the show, please consider sharing the episode, leaving a review, or donating to support our work. Thanks for listening. Before I introduce today's guest, I have a few announcements. It's been a while since our last episode, and the reason is that I've been focusing on finishing my Ph.D. Well, I'm excited to share that in April, my paper identifying a critical role for immune-acting fibroblasts, or IAFs, in skin inflammation and host defense was published in the Journal of Experimental Medicine, and I successfully defended my Ph.D. I'm excited to share also that the article was featured on the cover of the April issue of JEM and was also featured in a special collection. Stay tuned for a future solo episode wherein I go over the paper. With that, on to today's guest. Today, our guest is Dr. Miguel Reina-Campos. Miguel is a brand new assistant professor at the La Jolla Institute for Immunology in La Jolla, California. To quote his lab website, the Reina Lab seeks to understand the underlying principles governing tissue immune networks that enable robust and long-term protection against infection and tumors. Miguel completed his postdoctoral training with Dr. Ananda Goldrath at the University of California, San Diego, and earned his Ph.D. in the labs of Drs. Maria Diaz-Meco and Jorge Muscat at the Sanford Burnham Prebys Medical Discovery Institute in La Jolla, California. Miguel has published multiple high-profile papers in top-tier scientific journals, including Nature, Cancer Cell, and Nature Reviews Immunology. His new lab is using cutting-edge technology, including in vivo functional genomic CRISPR screens and subcellular resolution spatial transcriptomics to understand how tissue-resident memory T cells adapt to different environments, with the goal of identifying therapeutic targets for treating infection and cancer. In this conversation, Miguel and I talk about cancer, metabolism, tissue-resident memory T cells, spatial transcriptomics, advice for postdocs pursuing academia, and much more. I think you all are really going to enjoy this episode. Miguel is a rising star in the immunology field. He has an incredible life story and an incredible ability to teach complex biology. You can find a link to Miguel's lab website, his Twitter X handle, and references to the papers we discuss in the show notes. So, without further ado, please enjoy this conversation with Dr. Miguel Reina-Campos. Miguel, welcome to the show.
Thank you. I'm excited to be here. Well, I'm excited that you're here visiting LJI.
I'm excited to do this too. It's really great to sit down with you. And I want to start by getting to know you better, and if we could just start this conversation like every good superhero movie starts with an origin story. So can we hear the Miguel Reina-Campos origin story?
Well, I don't know if I'm a superhero, but I did come to the United States more than a decade ago now, although people still think I just arrived. It wasn't straight from Spain that I came to the United States. First, I traveled a bit and I went to Germany with my wife to study and finish my master's degree. At some point, I realized that I wanted to go to the source of where science was coming from, the science that I was reading at school, that I was reading to get ready for my exams and my training. And for some reason, I kept reading papers that came from Scripps, Salk, UCSD, LJI. I'm like, what is happening in San Diego? And coincidentally, I also was trying to learn how to surf. And so in narrowing down the places that I could do science and surf, I came up with San Diego. And so came with $2,000 in my pocket more than 10 years ago, and now I have $2,000 in my pocket. But I've learned a great deal, and I'm really happy I did move to San Diego. Yes, it wasn't initially very easy. Hit us some roadblocks, especially with the immigration system. Also, economically, was a bit rough at the beginning, but I felt very supported by the community, very engaged by the scientists that I was meeting, and yeah, I'm glad it worked out.
Let's rewind the clock even a little bit further. When did you first get interested in science?
Not many people know, but both my parents are scientists. My dad has a lab at the University of Barcelona. He's been a teacher of cellular biology for many years. And my mom, she is professor as well, and she teaches biostatistics. So something that I never wanted to do. And such. But they had a great impact in me in growing up, but not so much as people think about making me study, but in instilling me a sense of curiosity and wanted to know how things worked. And that, came from many experiences as a kid. But one of them, for example, that I remember fondly is just collecting insects and different animals that we just capture and I put in pods and try to classify, and then just to see how they worked, and then look them up under a microscope that my dad brought from the lab and I had at home. And so, yeah, I think that I'm following the footsteps of my parents. That's pretty clear to me.
So it was always science. There was never any other profession that you said you wanted to be when you were a kid.
I think yes. As any other kid, I consider being a lifeguard and a fireman and an astronaut. But
Yeah.
but eventually, I just wanted to know how things worked, and I was fascinated by biology and the unit of biology, the cell, and how that little thing worked. And sort of as a segue, became fascinated by cancer biology as that cell that can almost do anything. And more than seeing it as a disease, I saw it as an opportunity to study biology.
So yeah, we're going to talk about cancer today for sure, because that's definitely one of the major through lines through all of your research from your grad school to now. Did that interest in cancer that started very early on as well?
Yeah, it started creeping in at the beginning of my studies in University of Barcelona, and through personal experiences with family and friends that I lost due to cancer, in trying to think of how that little cell became from a normal cell that was living in harmony within a tissue with all of the other different cells to become a very selfish cell that just wanted to survive and put emphasis on its own survival rather than the survival of the organism, and how that is enabled by things that are happening inside mechanistically. That fascinated me, the idea that a cell would never die. And then I learned that scientists were working with cells derived from people decades ago, and those cells were still alive and dividing. And how is that even possible? And I had a very good teacher during my scientific training in Barcelona who instilled that sense of wonder in the cancer cell. And yeah, that's everything I wanted to do, I think, from my second, third year of college.
So another major through line through all of your research is metabolism. Did your interest in metabolism come after your interest in cancer, or did you also start looking at metabolism early on in your undergraduate career? Or it was master's degree also in Barcelona, right?
Yes.
Undergraduate and master's there.
Yeah, so I kind of used a master's degree in Barcelona as a conduit to being able to come to the United States. It's not easy for people in the United States to just send them an email and be like, hey, I'm Miguel from Barcelona. Do you want me to work for you? I'll work for free. If you do those kind of emails, maybe you get one reply out of 50. But if you go through a master program that you can say, hey, I'm Miguel, I'm on this master's. I don't need to get paid because I have a fellowship. And so that was easy. And no, I wasn't interested right off the bat on metabolism. Metabolism sounded like something obscure. Even today, I tell my colleagues I study metabolism and they don't really picture what I do very well. And they don't think metabolism is very important. But then I ask them, have you tried not to eat for one day? How angry you get, and try to not eat for 3 days. See how you do. And metabolism is actually the driver of what we do. Our whole life is geared towards where is our next meal going to come from. And I feel like cells also go through the same pressure. They need to make sure they are going to find the next source of nutrients. And that's such a powerful drive that I think shapes how not only normal cells, but also tumor cells behave.
So when I think of, and I am not a cancer expert or a metabolism expert, but I think what I think of when I think of cancer metabolism is probably what most people who have had a science degree think of when they think of cancer metabolism. That's the Warburg effect, right?
Yeah, and that's how I initially started as well.
And can you define what the Warburg effect is?
The Warburg effect is a process by which cancer cells upregulate their metabolism that does not rely on oxygen, in which glucose is fermented very rapidly to lactic acid. And that way, the cell is able to generate ATP very fast, ATP meaning energy. So that's a process that was discovered early on and a process that was observed to occur in tumors. Whether that is still, the consensus is not entirely clear, because we also have seen that tumor cells do oxidative phosphorylation, which is the alternative to glycolysis, or the end steps on the glycolysis, in which glucose doesn't end up being fermented to lactic acid but gets put into the mitochondria, and you can squeeze more energy out of a molecule of glucose. You do it in a much slower manner, but still it's more effective. So what we've seen now with better technology is that tumor cells can actually do both. They do the Warburg effect, but they still use this oxidative phosphorylation to some extent. So we are
within
now,
a single cancer.
yes. So we're now revisiting a little bit how we think about tumor cell metabolism. But I feel like I'm getting ahead of myself, and actually this is not the type of metabolism I was studying. I felt like this was a bit crowded in understanding how things were burned down and extracted energy from. What I wanted to study more was the connection of what you eat with what you are, because people don't often realize, but a cell has different components in them. More importantly, the DNA, which has the instructions for what they need to do and how they need to do it, and the epigenome, who's built on top of the DNA, adjusting those instructions for a specific context. And the epigenome is composed by many different marks, and those marks are sort of like building blocks, like Lego bricks put on top of DNA. So those Lego bricks need to come from somewhere. That's also metabolism. The nutrients that the cell has captured from the environment and things that are happening inside of the cell make the products that are put into the epigenome. So that is a link between metabolism and cell fate. So what a cell is going to do and how a cell is going to behave, that was more underexplored as I was doing my PhD studies. And something very interesting that I found is that tumor cells will change their metabolism not only to fuel their needs for energy, but they also change their metabolism to change the epigenome, so they become something else. And that something else, maybe it's not as sensitive to the current therapeutics, and then that allows them to avoid current therapeutics, and then you have a big problem.
So you said in the beginning there about what you eat. How does what you eat play into that process?
Yes, I well, actually, I don't know. That's what I'm studying, but I think there are three different levels when thinking about metabolism that sometimes people just put into one.
Mm-hmm.
So this is a long answer to that question. So just,
We got time.
Yeah, bear with me for a sec. So when I think about metabolism, at least now, I think of three different layers. The first layer is within a cell. What's happening inside of a cell? It could be an immune cell, or it could be an epithelial cell in your skin. And those are the enzymes, the substrates, the transporters, the chemical reactions that are happening for that cell to acquire energy and building blocks for proliferation and building their genome. That is in equilibrium with the second layer, which is the environment. And that is the metabolism that we think of accumulation of lactic acid in muscles after exercise, for example, or the tumor microenvironment and the buildup of toxic molecules and other nutrients. So those two layers of metabolism are obviously in equilibrium. And they are also in equilibrium with a third layer of metabolism, which is the organismal physiology. It is, did you just drink Coca-Cola and your levels of glucose are off the roof, and then the environment in your cells also has more glucose, now the second layer, and now inside the cell there is also more glucose, the first layer. So all of these three layers are in equilibrium and talking to each other, and there are complex mechanisms dictating this. But you also have to think that they are somewhat insulated, and they are insulated for a reason. It's because you don't want your intracellular glucose going super high after you have drunk a very sugary drink, because cells need to have a specific concentration of glucose, very well regulated. So these three layers are connected but also insulated from each other through some unknown mechanisms. And so when thinking about how the things that you eat affect your cellular metabolism, you need to take into account that there are going to be many processes insulating and buffering this thing. And so if you don't eat for a couple of days, I mean, you're going to be very angry, but your cells are not going to die because there are built-in mechanisms, for example, the pancreas is going to make some hormones that are going to release storage of glucose. So the intracellular concentration of glucose are going to be maintained, essentially. And so, yes, it will have some effect what you eat on how your different organs are working, but it's somewhat buffered. And that's for a reason, because we were not evolved to have a deli or a supermarket where we can just buy and get food. We used to have to go out and hunt and maybe starve for a couple of days before we got our next meal.
What you move on to in your postdoctoral work is a different aspect of metabolism than what you studied in your master's and graduate education, which is the metabolism of the T cell, the immune cell. So, if you're thinking about immunity as a war where you have the T cells versus the cancer, you were studying the cancer first. Now you're studying the thing that's going to kill the cancer. And I was wondering, hoping you could first just describe the type of T cell that you specialize in, which is the tissue resident memory T cell.
Yes. So again, maybe I'm gonna go on a little journey here for a sec, because the underlying connection between all my science has been this fascination for how a cell can adapt and survive to its environment. And as I said, I was fascinated by how cancer cells did that. And my focus was to understand that so we can, simply put, cure cancer, understand it. As I did my research on different signaling pathways and metabolism of the cancer cell, I obviously realized there is way more to it than a cancer cell in a tumor. And some tumors are actually composed by only as few as 10% or 5% of tumor cells. Everything else are just normal cells that have been caught in the crossfire. And those normal cells are also going to promote, some are going to promote the tumor, some are going to try to fight it. And thinking of things that we could use against the tumor cell, I realized we needed to engage the immune system. And as you very well know through your own research, other normal cells in that environment. I also realized that everything that I had learned about how a cancer cell works could be applied to the immune system. And the same things that I was trying to inhibit in the cancer cell, maybe I could try to enhance in the immune system to give it more tools to fight the tumor cell. So through studying my enemy, I was able to go and enhance our friend, which is the immune system. And so in thinking about what part of the immune system I could focus in, I was fascinated by learning that there is a new subtype of T cells that were discovered no longer than a decade ago. So around the same time I came to San Diego, which is kind of hard to think, but many different labs sort of at the same time realized there is a population of memory T cells. These are T cells that are going to protect you against reinfection because they've already seen the threat. And after they've cleared this pathogen, are going to differentiate and go to every single tissue in your body and are going to establish residency. So these T cells, what we call tissue resident memory T cells, make a home out of the tissue and they never leave. So a T cell that might go into a lung to clear an infection, then when the pathogen's cleared, that T cell will stay in the lung for as long as a decade or 15 years or more. I mean, there are some studies looking at transplant donors where they've looked at the organ of a transplanted person many years after the transplant, and the original cells in that organ were still the T cells from the donor, meaning those T cells never left and never died. So in thinking of ways to leverage the immune system against cancer, knowing that one of the limitations currently is the poor persistence of T cells in solid tissue environments, my idea was, okay, let's just learn from a professional survivalist, professional T cell that is able to stay for years in a tissue. And let's see what secrets they have, and let's see if we can translate those to the next generation of immunotherapies.
A lot of listeners, I'm sure, because of the COVID-19 pandemic, are familiar with B cells and T cells and a little bit about viral infection, which is a model you also use to study the tissue resident memory T cells. Could you describe the sequence of events that happen after you have an infection that lead to the accumulation of these tissue resident memory T cells in your tissue? And If there's gaps in there where we don't know how things are happening, that's totally fine. I'm just curious if you could delineate like the sequence of events.
Yeah, so I didn't know about this myself much, and I joined Ananda Goldrath lab at UCSD, who is an expert in this field. And what she told me is that T cells at the beginning, when they are naive, they haven't seen a threat, they get activated by an antigen presenting cell. They go to a tissue based on the instructions they have received. And these are instructions that we still don't understand very well how they work. Because if you can imagine, you have an infection in your gut and you have a naive T cell in a lymph node somewhere in your body that's being primed by an antigen presenting cell. That antigen presenting cell should tell the T cell to go to the gut and not go to somewhere else, right? So those signals are still not very clear, and we're just starting to understand better how that works. So the T cell is deployed to a tissue, and that T cell has already seen some instructions knowing, oh, I'm going to the gut. I should upregulate some molecules so I get better adapted once I get to the gut. It's a little bit like they get to your home and they're like, hurry up, you're going, you're leaving, you're taking a plane to, and they tell you where you're going. So based on where you're going, you start packing specific things. But also you don't have much time to pack up your things. You really need to go and get there. So it's a mix between being pre-programmed for a specific tissue. And once you get to the tissue, those T cells obviously have to fight the pathogen. And then in my mind, that's how I imagine the T cell has entered a tissue, which we don't really know how they do that yet. We don't know if they enter through the different architecture of the tissue, through specific areas or not. But based on how they get there, then in my mind, they look around and they start sensing some specific environmental signals. This could be cytokines or this could be nutrients. And based on that, they make decisions. Am I going to stay or am I going to leave? And also a little bit based on the capacity to stay, if there is enough room for them, then they start a process of differentiating, meaning they change how they behave and a little bit how they are. So for example, if they get to a place where there is no glucose and there is an overabundance of lipids, probably these cells will reprogram their metabolism a little bit so they can make use of these new environmental lipids. But if they get to a place where there are no lipids and only glucose or amino acids, maybe they want to reprogram their metabolism a little bit. So in thinking of a cell getting into a new environment, I kind of feel like when I came to the States, right? You get here, I was pre-programmed to come to the States, but it wasn't until I was here that I knew I needed to get a car as soon as possible. That was my first formula of adaptation to American lifestyle. Also a credit card. And so I started learning the rules. So I feel like T cells do that process of differentiation, which is a continuum. It doesn't really end. When a T cell infiltrates the tissue and everything's been cleared, they start this process of differentiating towards a tissue resident memory cell that is going to offer this protection long term, but it's never a process of, okay, I've made it. Just like you and I, you never really make it. There's always something. And so I think these T cells are also differentiating continuously within a tissue, changing their location either slowly or fast, depending on how they have to survey that tissue. And they become acquainted with the place and very well adapted.
It's like an immune outpost, kind of stationed where the war is happening against the cancer or the virus.
I kind of, yeah,
It'd be like a first line of defense, right?
Exactly. I like to think of that way. And some tissues is very clear that that's that's the case. For example, the intestine, where you see millions of T cells that are tissue resident packed within the villus that are also uptaking nutrients and protecting us from pathogens that want to get through the epithelial barrier. Other tissues are not so dense in their immune content, in homeostasis, like the liver or the kidney, but they also have tissue resident memory cells. So in thinking of what controls the density of immune cells, is there a maximum capacity for a tissue? And is that capacity expendable? What expands it? We know tumors are also very heavily infiltrated with immune cells initially, but then become immunodeserted. So immune cells don't like living in them and then become excluded. If we understand how some tissues are able to host so many immune cells, maybe we come back to a tumor that's immunodeserted and prevent immune cells from being excluded.
So when you have an infection or cancer and you have that antigen presentation and differentiation and priming of your T cells, there's also other types of T cells, right? That
Yes.
other than the tissue resident memory, there's the circulating memory, the ones that are just in your bloodstream, right? There's effector memory, which, and correct me if I'm wrong, but patrol the tissues, but they will also go back into circulation.
Yes. So sorry, I skipped that part. It's the name of the game is diversification for the immune system. So when the immune system detects a threat, it's going to diversify its arsenal so it can, cover all its bases. And the way it does that is by generating many different types of T cells that are going to recognize that threat in the future and then deploys them differently. So the same way that it generates tissue resident memory cells that stay as an outpost, as you said, it also is going to make effector memory cells that are going to recirculate in and out of tissues, like kind of doing a quick check-in, going out back into circulation. Is also going to make central memory T cells, which are mostly in the lymph nodes and also circulatory. And those would be like the headquarters for the immune system.
Do we know like say you get an infection and your immune system's going to make 100 T cells, say, and it decides to put 25 into the tissue resident memory bucket, 50 into the central memory bucket, and 25 into the effector memory bucket. Do we know what factors are controlling how the immune system decides how many tissue resident memory cells to deploy versus effector memory versus central memory?
That is a great question. The honest answer is that we still don't know exactly how that happens. We suspect that is linked to the other signals that a T cell is sensing while these instructions are being passed after an infection. And we have some clues that some cytokines, inflammatory cytokines, can do that. So if you have more of an IL-12 or IL-15 polarized response, you might generate more TRM than if you have lower amounts of IL-12 and IL-15. But the other many cytokines and how they play a role in allocating all these resources into different buckets, we don't really know. And it's a very important question because if you, for example, want to vaccinate, so you're mimicking a response and you want to generate tissue resident memory cells in the lung, which are the ones that protect you from actually catching that infection again, then you want to generate TRM. You want to mimic that environmental cue that are going to make those type of T cells. So if we want to make better vaccines, we need to understand those signals much better.
In the context of a cancer, say colon cancer, are tissue resident memory cells, T cells, the best T cell? Do you even need the other ones?
Yes, obviously, I don't want any colleagues to get mad at me, but I think the TRM are the most important, obviously. No, but
like for a systemic thing, maybe you need the others more, right? But if the problem's in the tissue, you probably want the T cells that are in the tissue, right? That are specialized to work in that tissue.
Exactly. And that's what fascinated me early on. The idea that there is this professional survivalist that can do all these many things. And also the fact that T cells are in tumors that have characteristics of tissue resident memory cells. And I want to be very clear that when there is an ongoing tumor, you can't really talk about memory T cells. So these are T cells that are specific for the tumor that are going to go inside of the tumor. They never really are memory, but some of them have tissue resident memory characteristics.
Why are they not memory?
So there is an ongoing threat. You can't really say they are memory because the pathogen, the
threat, they never had a time to relax and be re-challenged per se.
Exactly. They never had a time to cool off and generate what we call memory T cell after the threat is gone. Mm-hmm. So the inflammatory environment still is present and antigen is present. So these T cells are chronically activated and they are not allowed to become memory T cells. Mm-hmm. Despite of that, some of these T cells are inside of the tumors that can present some of these tissue resident characteristics because of surface markers and functional behavior, which, by the way, some of the pioneer research was done here at LJI by the VJ lab. These T cells that had characteristics of tissue resident cells are much better tumor killers, obviously. And they also are linked to better responses to immune checkpoint blockade. And I mean, it all makes sense. These T cells are better adapted and they have intrinsic yet unknown tools to fight tumor cells. So that is the underlying driver of my curiosity to understand these cells much better, because there is something inside them that make them very good and that will be very beneficial to know if we want to enhance current cancer therapeutics. I suspect that some of that is because they are metabolically adapted to a tissue environment.
Right. And in your postdoctoral work in the Goldrath lab, you did some very cool work investigating the metabolism of these tissue resident memory cells using in vivo CRISPR screen. And I'd like to talk about that technology really quickly because it's really pretty awesome. So I'm going to see if I can recapitulate it here. So you use, first of all, a special type of mouse, this P14 mouse, right? That is as T cells, CD8 T cells that are specific to this LCMV virus, correct?
Correct, yes.
And then you adoptively transfer or transplant those cells into a congenic mouse, right? Which means we have different markers on our cells like the CD45.1 and CD45.2, and you transfer in the LCMV specific T cells from the 0.1s into the 0.2s. And so when you use antibodies, you can stain for CD45 and see which ones are the donor versus the recipient. And so that's the model that you use, right? But in this particular experiment, you did a pooled CRISPR knockout approach on the T cells first, right? Before you transplant them into the recipient animal. And you were targeting with that screen metabolism genes, right? The question was, does T cell metabolism affect the tissue resident memory cell response to LCMV? Is that correct?
Yeah, it is. So in essence, some of the tools that we use are specific T cells that have specific TCR. So we know their specificity is towards some of the epitopes in LCMV. We use LCMV because it's a acute virus that has a systemic infection, and that way it generates TRM pretty much in every organ that we've looked. And because we have one clone, this P14 T cells, that we know react to LCMV, in a way we are able to see tissue resident memory cells in every single organ in a mouse in a manner that we control over time. So what we do is infect with LCMV and then transfer in to the mouse our P14 CD8 T cells. Those P14 CD8 T cells are going to recognize LCMV and mount a very synchronized immune response. And that way we can follow, using our congenics, as you said, the CD45.1, transfer cells into a CD45.2 congenic host. And we can, in much detail, just follow how these T cells differentiate and go to different organs and generate tissue resident or effector memory or central memory cells, and follow this continuum of differentiation over time. And so the question that we had was, what are the different metabolic adaptations of T cells to different organs? And how do those compare to T cells that just live in circulation? We started doing that by just looking transcriptionally if T cells are different in different organs as they relate to the expression of metabolic enzymes. And the first results were strikingly clear that T cells that live in different environments have different metabolic profiles transcriptionally.
Was the interest in metabolism just because you were interested in metabolism before, so you're like, I want to see what metabolism is going on here? Or was there an indication that the tissue resident metabolism would be somehow important or different?
I mean, yes, this is based on previous observations by Laura McKay and others, that they've seen that tissue resident memory cells living in different organs have expression of different molecules related to fatty acid binding proteins. So that was one clue that I need to acknowledge to Laura and others, that they already had suggested metabolism was going to be different. We tend to do things we're comfortable with, and I knew metabolism played a big role in T cell activation and differentiation, and why not adaptation to different tissues? Based on my previous experience, I know that metabolism of the liver is not the same as the one in the intestine or the pancreas or the kidney, and obviously not the same one you have in the blood. So to me, it was a low-hanging fruit that nobody really had asked yet, how systematically addressing the different metabolic adaptations of these T cells. All these bioinformatic initial analysis pointed towards very striking differences in pathways, for example, related to hypoxia or making lipids or digesting glucose. So it was a matter of just now functionally testing all these different metabolic enzymes. And I couldn't have done this with amazing collaborators that I had during my postdoc. And Hongbo Chi, a great PI at St. Jude, had just published a paper using a metabolic library that allowed to screen more than 3,000 metabolic enzymes in T cells. So I asked Hongbo, could I use your library? And Hongbo said, of course, here it is, which I thought was extremely generous. And so the only thing I had to do was put this library into T cells, put these T cells into the mouse, infect with LCMV, and then asked how the different knockouts of the different metabolic enzymes affected the formation of tissue resident in different tissues.
And the punchline to that story was that dietary cholesterol was not so good for tissue resident memory T cell development in the gut.
Yes. But for some reason, they looked like they wanted to make cholesterol. So that was a head scratcher for a long time. Why would a T cell upregulate the entire pathway, almost all the entire pathway, to make something they don't really like? Yeah, that was, I think, 2 years of my postdoc trying to figure that one out. And then the answer came by looking at how every single step on that pathway impacted the formation of those cells. Because I had the functional data from the screen, I could go back and ask, oh, is the step 1 important, yes or no? Is the step 2 important, yes or no? And it turns out that the further I went on that pathway, the lesser important were those enzymes. By the way, for listeners in thinking about how a metabolic pathway works, a substrate becomes a product through an enzyme, and that product is in itself a substrate for the next enzyme. And so it's a chain of reactions. In this case, around 25 reactions. And so what I realized is by reaction number 14 or 15, T cells no longer care if I remove that enzyme from them. T cells living in the small intestine in this case. So how is that so? What was happening? So it turns out that products from this pathway are also feeding adjacent pathways that are going to make very important downstream metabolites for T cells. And so once I picked up on that clue and realized that T cells were not trying to make cholesterol, they were trying to make other things, then everything started to become much clearer. Now, what other things are they trying to do? That was a bit complicated to figure out because there is not one additional product that can come out of this pathway. There are multiple. Some of them are just important for all the T cells, like there's an impronounceable name called geranyl phosphorylation. Geranyl. That's why. Yeah, it's a complicated. Geranylation.
Wow.
Yes, it's complicated. So that one is important for all the T cells. So that wasn't really explaining why T cells in the gut specifically care so much about this. There's other products with all more difficult names even, but there's one thing that I realized was very specific for T cells in the gut, and that's called coenzyme Q, also known as ubiquinone. And that is a lipid that lives inside the mitochondria, and it's very important for transporting electrons from one complex to another in the electron transport chain for making energy. And by the way, you can buy these as a supplement.
Right. A lot of people take that.
Yeah, a lot of people take that. So one question I had is, can I just give mouse infected with LCMV this supplement? Will they do better? The answer is no, unfortunately, because there's huge issues on the uptake from the diet for the T cells in the gut at least. So maybe that's one reason why they need to make it. It's not so easy to uptake from the diet. And so these T cells have an increased requirement for making coenzyme Q, which in turn is going to charge their batteries, meaning the mitochondria, the power hub of the cell. And that's good for them. And the hypothesis is that it's also going to be very good for T cells trying to fight a tumor. As you can imagine, if you are a T cell trying to fight this shifting, nasty mass of cells that you need to kill, you're going to need a lot of energy. And so once I had that breakthrough using infection models in which the system works well, I could translate that into context of tumors, and specifically colorectal.
So you see the detrimental effect of dietary cholesterol because it's depleting the batteries of the tissue resident memory cells? Is it causing the reverse to happen in them?
Yeah, so metabolism is a bit like a flam. You push it on one side and it moves, right? It adapts. So in every metabolic pathway, there are feedback regulatory loops. For this specific pathway, it's very clear and it's very well known that more cholesterol is going to shut down the production of internal cholesterol. As you could imagine, if the cell senses it has enough cholesterol, it's not going to make it.
Even though they're not even trying to really make it.
Exactly. So that's the trick, right? If a T cell that has to make intermediates of the pathway senses it has the final product, it's going to shut down the entire production.
Mm-hmm.
And so it's also going to shut down the production of the important intermediates. That's why it was so complicated to figure out.
So remind me of the experiment, because you did a dietary cholesterol experiment. Do you do that in the context of the cancer, or was that just the LCMV infection?
That was just in the context of LCMV infection.
Do you think it would work in the cancer too, where you give more dietary cholesterol, you have worse anti-tumor immunity, and then maybe the reverse is true too, where you lower the dietary cholesterol because then the T cell will think, oh, there's no cholesterol here, we need to turn up the synthesis. We can have better anti-tumor immunity.
That's an excellent question, and that brings me back to when I was talking about the different layers, right, of the metabolism, what's happening inside, layer 1, what's happening in the immediate environment, layer 2, and what's happening in the physiology, your diet and your exercise and so on. Because if you just eat a lot of cholesterol, there are ways that your body is going to dampen the massive intake of cholesterol, unless you're a T cell living in the intestine and you're just extremely exposed to that influx. Okay, so if it's not a diet, how can we do it? And I think the answer is just by going and finding specific enzymes and proteins inside of immune cells that we can manipulate, that we can manipulate through drugs that we can give people and they're going to specifically target immune cells, or cellular therapy where we can get the immune cell out of somebody's body and genetically modify it and put it back in an enhanced form.
So we're talking about just these cells at first and how we're breaking up the immune system into different types of T cells. And some of your more recent work, your bioarchive preprint, is now doing what immunologists like to do and breaking things up into even more subgroups. And you've identified, using some more really awesome technology, different types of tissue resident memory T cells. Can you tell us a little bit about the technology that you used in this paper? And you used very similar experimental models, right?
Yeah, and I do feel the sentiment of immunology is trying to subdivide subsets into more subsets. But I don't think that was per se our objective here. And
it's a necessary thing. I'm not disparaging.
No.
It.
It's fine.
We need to define things.
Yeah, we have
to understand them.
But I think it goes against us, and we need to be aware of it. And yes, what we sought out to discover or to prove is we know there is diversity in a T cell response, as I said, that diversity is seen by differences between organs. A T cell in the liver is not going to be the same as a T cell in the small intestine. But also even within the small intestine, T cells don't all look the same. We know that there is diversity as well in that compartment. And that diversity is functionally very important. Some cells are more effector-like, so they're going to maybe kill more. And some cells are more stem-like. We're going to have more of a precursor role in replenishing the pool. And so the hypothesis was very simple. Are these cells different because they are in different places? And if they are in different places, what's dictating that diversity? Is it because they're interacting with different cells or sensing different signalings or cytokines? And this had been very hard to do until 3-4 years ago when spatial transcriptomics came along and allowed us for the first time to look at single cells in tissues in their natural environment, and at the same time as we could look at their transcriptional profiles to look at their differentiation. I think now for the first time we have the capacity to allocate specific functionalities and phenotypes within immune cells to specific micro-niches. And I don't know if people are picturing a tissue, but within, for example, a small intestine, you have this finger-looking structure that's like the villus. And at the top of the villus is typically where absorption of nutrients happens. And where enterocytes are absorbing all these nutrients, at the base of the villus, there is this crypt where stem cells are making more of this epithelial barrier. And you'll find different immune cells that you see at the top. So the conditions and the environments are going to be very different within this very specific micro niche. And we found exactly that. We found that T cells that lived on top of the villus look different than the ones at the bottom. And there is a continuum of differentiation occurring after infection that takes T cells from the bottom up to the top. And we were able to find some of these signals and cellular contacts that dictated that diversity. And to us, I think that is a bit of a breakthrough, because you can see reflected on scientific illustrations all over the place that people will just draw a small intestine or a tissue and it would draw immune cells kind of floating inside. And that's not how the immune system operates. The immune system is very intricate, complex, elegant system that has found the best solution to accomplish a very specific task, which is protect you from becoming infected. And there is a reason why a T cell needs to be in direct contact with an epithelial barrier. We still don't know exactly the reason and how they do that, but they are in that specific location for a reason. And so in thinking about finding better therapeutics or better vaccines, we need to understand how to deploy those T cells to those specific environments.
So I want to talk more about the technology, the spatial transcriptomics. You know that I'm very interested in this. Can you describe the different types of spatial transcriptomics? And you used several in this paper. Can you share with our audience which one you are thinking is going to be the most valuable going forward? I know you've invested in a system for your lab that you're starting currently. Can you tell us why you picked that one and more about how you're thinking about spatial?
Yeah, so for those who are listening, spatial transcriptomics is a technology that allows us to look at the transcriptome, the mRNA molecules that compose a cell in tissues or in space. And that is done through two different ways. You can sequence the mRNA, the transcriptome, or you can use probes that are going to directly target. So it's either sequencing based or optically based. Sequencing based, that's what initially opened the field to whole transcriptome, more comprehensive analysis of the transcriptomes in situ, but also lacked from the resolution that we needed to capture our immune cells. I don't want to name names because they are not sponsoring this podcast, and they haven't sponsored much of my research either. But there are some companies.
We'll take any sponsors if anyone wants to sponsor us, though.
Yeah, they don't need more publicity. And so these companies have developed technologies that allow it to capture full transcriptomes in situ at resolutions going from 100 micron to, I think now, 2 to 8 micron, but still are not what you would say single cell. But they do have almost the entire transcriptome. So these are very nice. And I'm actually obviously very grateful that these technologies exist and for these companies to put them out there for us to use it. And then there are other technologies that are based on capturing specific mRNA molecules through probes. And those are technologies like MERFISH or rolling circle amplification that have been the basis or the chemistry that some companies have used to develop their own instruments. And those techniques allows us to look at between 350 to now around 5,000 genes in situ at single cell resolution, actually with subcellular resolution, because you're actually just seeing single mRNA transcripts. To me, that was a breakthrough. Being able to observe that immune cell in its unperturbed environment and seeing, oh, this cell has this transcription factor that's going to make it more tissue resident, or it's going to make it be able to capture a lipid, or do all these kinds of things, and just look at it in the context of all the other transcriptomes and neighboring cells. So I think that has enabled research that I didn't even dream to do 5 years ago. It also has given us a lot of headaches in how to analyze this data, as you know, tremendous amounts of data that are hard to process. And I want to mention that the community around spatial transcriptomics is extremely supportive. It's a field that's blooming. I'm not a computational biologist, so I've always relied on extremely smart and generous collaborators. The colleagues I have in my lab before I started my own lab here, who have enabled all these discoveries. And so in thinking what is the best technology moving forward, I honestly don't know, because this field moves so fast. But in my mind now, there is not one technology that does it all. So at least moving forward for the next, I would say 6 months to 2-3 years, the best approach would be a combination of hypothesis generating whole transcriptome, even if it's a slightly lower resolution, with a very highly resolved, maybe targeted probe design. So combining both sequencing based for discovery and probe based for hypothesis testing and better resolution. And obviously the devil's on the details, right? What technology you can use to do that? What's the chemistry that supports that? Does it work for FFPE? Does it work for fresh frozen? What's the surface? What's the actual resolution? What's the size of the actual raw data that you get? Can you actually handle all of that? But yeah, I'm excited that I've seen this field bloom, and I had the privilege to be able to work on this almost from the beginning.
So I want to get a little granular here and talk about some of the specific analysis that you did in your bioRxiv preprint, because I thought it was very inspiring. I've seen a lot of spatial papers coming out recently, and a lot of them are atlases, and they kind of just tell you, okay, this is where stuff is. But in your paper, your colleagues did an in-depth analysis and used approaches that I have not seen done before. Can you tell us a little bit about what you did with those data, aside from just saying, okay, here's our clusters in our UMAP, and then here's where they are in the tissue, and that's that? What else did you do?
One of the first things that we did, and I think that actually what took us the longest, is thinking about a spatial framework we could use in order to analyze our data. Because you're looking at a tissue that's very complex, and then you see, oh, things change, right? And then you describe those changes. But how do you describe those changes in a way that you first understand, and then other people will understand as well? So the small intestine was sort of an easy first entry into this problem, because the small intestine has its functional unit very well described, the villus and the crypt. And so this unit is repeated all over the small intestine, from the proximal part of the intestine, contact with the stomach, to the colon. And so essentially, by overlapping all the different villus on top of each other, we could generate a coordinate system that's based only on two numbers. The first coordinate is the distance of every immune cell to the nearest epithelial cell. And the second coordinate is the distance of every immune cell to the base of the muscularis. And so that generated a 2D plot, which as immunologists are comfortable with, because it looks like a flow plot.
Yeah, it looks like a flow plot.
And so something that I can, in 5 seconds, you're looking at that plot, which we called immune allocation plot. And that's an IMAP.
A play on the UMAP.
Yes, a little bit. And that's something me and my wife came up during a hike that we did here in San Diego. But in essence, that IMAP, what tells you in one quick view is, are things changing based on functionally important architectural unit of the tissue. We hope we can make more IMAPs for other tissues, but that's a different story. And so what we did first is to observe after infection how T cells changed in that IMAP space. And what we saw is that T cells initially are at the bottom and then slowly move up the villus. I'm not saying T cells do that, but the majority of the population, what we saw at the beginning, the distributions changed, right?
Over, because you did a time course too,
Yes.
So you can see the distributions moving on that IMAP plot.
Exactly, right? So now we could find molecular correlates of the distribution spatially to the differentiation of the T cell.
Right, so like differential expression based on the distance these things are in the IMAP and that
Exactly.
coordinate system.
So, for example, for T cells in the small intestine, something very important that needs to happen is the upregulation of retention molecules that are going to allow these cells to stay in the tissue. For epithelial surfaces, that's CD103, for example. And so T cells in the small intestine need to upregulate CD103 in order to become fully differentiated tissue resident memory cells. And we observed that T cells that are upwards in the villus have higher upregulation of CD103. Now, can we perturb that system? Can we prove that's cause and effect? So we knock out the receptor for TGF-beta, who is the responsible for upregulating CD103, and we observed that T cells no longer went up the villus, and they had a limited capacity to upregulate CD103, as has already been described. So that's just one example of how first we showed our system works, and we're able to find these differentiation differences in situ, that we could perturb the system, and the system behaved as we expected. And so looking forward than that, or looking beyond that, we started looking at whether there were gradients and differences in signaling. And it was so striking. It was actually very hard to find a gene that wasn't spatially dictated somehow. And in looking at these two main axes, the crypt-villus axis, so the length of the villus, or the nearest epithelial cell axis, we found many cytokines and chemokines that were differentially expressed. So these chemokines and cytokines that immunologists have known for decades, they are extremely important. It turns out they're extremely polarized in this particular environment, like IL-7 or IL-10 or IL-15 or IL-21. And we, for the first time, we could just look and know where those cytokines were coming from, something that immunologists have been wondering for decades. And so we found a very exciting gradient. This is not in the bioarchive, by the way. This is sort of like something that we just found.
This is the inflammatory content exclusive, right?
Yeah, so this is something that we're working on as part of the revision that we have for this paper. But we have found a new, it's not a new chemokine, but it's something that's very highly expressed in the lamina propria. So this is in the middle of the villus, and it's not so much expressed in the epithelia. And so you get an idea. This is like maybe 200 micron worth of distance. So maybe there are like 10 to 15 cells making this gradient. So it's very small. Only with this type of technologies that allow this resolution, we would have been ever able to see this. But this gradient, although small, is very important for how T cells are distributed between the lamina propria and the intraepithelial layer of the epithelia. So going back to the functionality of where T cells need to be in order to do their job, right? Do they really need to be in proximity but in contact with the epithelium in order to protect from a pathogen coming in through the barrier? So we perturb the system using a new CRISPR approach that we can detect through spatial transcriptomics. And indeed, when we remove the receptor sensing the chemokine coming from the lamina propria, now most of these T cells were distributed in the intraepithelia. So in short, what we found is a receptor on T cells that is allowing the cells to become positioned in the lamina propria. And that goes hand in hand with their differentiation potential. So in essence, what I think we have proven is that the identity, the differentiation of T cells is intertwined with their location in a tissue. These things go hand in hand. Now, when I think about fate specifying factors, I actually think about fate/location specifying factors. Because if we can send T cells to a specific area, we're probably inducing a specific differentiation profile. If we can induce a specific differentiation profile, it's likely those cells will end up in that area. So it is something that we need to think more and more about how we manipulate the immune system, and thinking about these T cells do not operate in a vacuum, and they are many different complex cellular contacts and sensing of cytokines and nutrients that are going to make a T cell in a tissue.
This paper that you have in bioarchive is focused on LCMV. Your previous paper in the Goldrath lab was LCMV, and then at the end, cancer, right? You don't have any cancer in this paper, but I imagine you're working on it. Do you have any suggestion of where those T cells might be, the effector versus memory in a cancer versus LCMV? I don't know much about LCMV, but are the epithelial cells at the tip of the villus, are those the ones that are infected, and that's where the tissue-resident memory cells that are effector need to be to clear them? Is that why they're there?
Yeah, that's a great point. My understanding was that LCMV mostly infects cells that are lower in the villus. And the epithelial layer of the intestine clears up very fast. It's renewed every 2-3 days. So I think the cells that become infected are somewhat lower.
But they move up, right?
But they move up. Yeah, I don't know.
Maybe it's just there with its mouth open like a hungry hippo.
Yeah,
right?
yeah. So that's another thing that when thinking about these T cells, we see them static in a villus. They are actually on, those mechanical stairs, sort of, because the epithelial cells are sort of moving upwards as they get replenished by the stem cells. So the T cells that are tissue resident and don't ever leave, it's not like they are static, just poised. They actually, in my mind, they need to be walking back a little bit. But in tumors, I think there is going to be definitely a spatial component to it, and we are following this up in my lab here at LJI, is in thinking of a tumor as an even more complex environment that's also changing faster than healthy tissue at homeostasis. And we've done spatial transcriptomics using models in which we can follow also T cells that recognize specific antigens on the tumor. And we do see that their differentiation and functional capabilities are different depending where we look inside of the tumor. When we look at T cells that are outside or next to the stroma, they display specific markers than those that are inside, sort of fighting the cancer cells head to head.
I imagine the ones that are inside are going to have that more effector memory phenotype versus the one outside. It's going to be more of the memory phenotype, right?
Exactly.
Do you think, and I imagine you're going to test this, if the TGF beta is playing the same role there, if you inhibit TGF beta, do they stay outside the tumor?
Yeah, this is very well known, but it is known that TGF-beta is going to play a massive role in controlling immunity in a tumor environment, and that's research done by, well, I'm blanking now on her name,
We'll put it in the show notes.
a fantastic PI at Genentech, and others that have looked at how TGF-beta influences this. I think that's honestly a bit crowded and not solved, but on the way to being solved. And what I want to do now is thinking about what are the cellular contacts and what other things that are going to promote the existence of these more stem cells within tumor environments, and what is going to promote the acquisition of tissue resident characteristics by some of these T cells that are inside of the tumor. So again, looking at some specific markers like CD103, which we know labels T cells with superior intrinsic ability to kill tumor cells, see if I can find them inside of a tumor and see if I can find out who they are interacting with. Maybe there is another cell telling this T cell that it should upregulate CD103 and equipping this T cell with better anti-tumor properties. Whether that's a cell contact or whether that's sensing specific signals like cytokines or nutrients, we don't know yet. We're starting to look. It all points to a combination of both things.
So you have this paper now in bioRxiv, and you had the big Nature paper before that, and you went on the job market, and here you are now at LGI. We're in your office, your brand new office. It's really nice. And I want to talk a little bit about getting the job. I'm sure some listeners are graduate students or postdocs, and some may be applying for jobs now. Can you describe a little bit or share some words of wisdom for listeners about starting your lab? How did that process go? Some of the challenges. And we could also talk about what you're going to focus on too with your lab.
Absolutely. It all feels like a blur, honestly, in which it was a...
Congratulations.
Thank you. It's been a while, right? I'm very fortunate, and I think that also speaks to my past experiences in San Diego, because I came to San Diego a decade ago, as I said, and I never really left, but I also never had the need to leave because I felt the community and the scientific field was just bubbling up. It's just more and more and more and more, and it never really saw signs of this slowing down anytime soon. After my postdoc with Ananda at UCSD, and you would say my success as an academic, then I started to think finally on, okay, I want to set up my lab and I need to go into the job market. And so I had the privilege to attend some conferences and training symposiums on how to become a PI and how to work on my management skills, on thinking about mentorship, or what makes up a good mentor, what makes a good mentee, and my own background of a bit of a struggle in coming to a new country as an immigrant. And so when I decided I wanted to go into the job market, I focused on 10, 15 places I really wanted to work at and prioritize.
Where you could serve.
No, actually, prioritizing the science.
Oh, wow.
And I realized, yeah, your priorities change and everybody is going to be different. Some people will have kids and some people have partners that maybe need to live in specific areas of the United States or elsewhere, and that's going to limit their choice. In my case, I was in a position where I could essentially think where I wanted to do science and who was going to enable that science the best. And what are the chances that that place was just a 5-minute commute from my other work? I mean, very few places in the world that happens. And so, yeah, my idea was, now that I negotiated my offer, I can say this, my idea was to stay here in San Diego all along. But I did go and interviewed and met amazing investigators at other places like U Chicago or Princeton or the Allen, where I could just talk to many different people and they gave me great feedback and made me think even deeper about my problems, my scientific problems. And then coming to LJI to expose my work, I was much better prepared. So that's my advice to some people that are now in the job market is don't necessarily limit yourself to where you can go. Just see if you can get as much exposure as you can get. That's going to be excellent training for when you get your dream opportunity to really shine. And so I'm obviously thrilled that LJI believed in me and I was able to stay here. And in thinking about the future, I'm excited that LJI is embedded in the bigger San Diego community. And not only that, the scientific community, I feel like in immunology especially, is extremely welcoming. to a new investigators. Silvio Gatkin put it recently on an immunity opinion article where we are immunologists by accident. As I said, I didn't start being an immunologist. I was a cancer biologist that saw an opportunity in studying this. So I also, like Silvio, define myself as an immunologist by accident. But the community has been so welcoming in teaching me new things and being able to collaborate, like Hombo, like Laura, and many other PIs. And so, yes, that is in essence my advice. So just get out and talk to people and get exposed.
We talked about before we got on the air here about the name inflammatory content, and you were wondering if there was going to be any controversial topics discussed. And I got a question for you. So,
All right, should
it's have you heard of the term hot take before?
Oh yeah, of course. Yeah.
So, in terms of advice for trainees or bad advice, do you have a hot take? Something that you believe in terms of applying for jobs or how to be a good scientist that's controversial?
You need somebody on the inside.
You need somebody on the inside. What do you mean?
Yeah, it's extremely hard to get a job if you don't work your personal network.
And you think that's true or that's false?
I think that's true.
It's true.
That's my hot take.
Okay.
I think that
You need help. You can't do it alone.
Yeah, I mean that you need help and you need to work on your personal network to get your dream job. I'm sure there is people who just out of the blue were extremely amazing, extremely amazing, and they applied somewhere where never ever heard of them, and they were immediately impressed and they got a job. But I honestly don't know any of those people. Every node that has made it to their dream job, it's because they checked all the boxes. They are not hiring you only for you as a scientist, they're hiring you as a colleague as well. And a good way to show that you're going to be a good colleague is that if you previously had developed your own personal network of relationships. It's not about favors or owning favors. It's about going to conferences and being a good collaborator and being a good mentor and being open and willing to discuss science with all kinds of people. And if people who are considering you as a prospective PI in their institution, they will know that. And maybe you have 1, 2, 3 friends in common that you didn't even know. And maybe because you asked that question at a seminar 2 years ago and the speaker reminded your name, or maybe you spoke with him during lunch, maybe you went to their poster, and that person now is sitting in your recruiting committee and they remember you from there. And so that's why I said you need someone in the inside that wants to vouch for you, because this is a competitive market and there will be hundreds to hundreds of applications.
You had another hot take that you told me last time we spoke. I'm going to bring it up now.
Okay.
You said, don't worry about a K99.
Yes, the K99 system is broken.
That's a hot take.
That's a hot take. Yeah. Okay, I'm going to go with that one too. The K99 system is broken. Okay, I said it. It just doesn't make sense. It just doesn't fund the best science. I'm sure there are great scientists being funded by K99s and it worked for them. I actually admire them a lot because writing a K99 is the hardest thing I've ever done. More than any revision for a paper that I've ever done, because you have to work at the same time on your science. For those that are not familiar with a K99, you have 4 years to get a first author publication and apply for a K99.
During your postdoc.
During your postdoc. So imagine somebody who just moved country and starting in a new field. And now you get to a place and then you're developing a new project and learning about a new field. And you have 4 years to apply for a K99, which they don't say it's a specific strict requirement, but you do need a first author publication to get a K99 for the most part. Now, how are you going to do that in 4 years? It is impossible. I'm sure people have done it. Maybe they found a project that was already up and going, or they were extremely smart and successful and they make it work. But that's such a minority. The K99 system is broken.
I think the intentions are good, right? The intentions are to get people to not stay in a postdoc forever, but maybe 4 years is too quick, you think? You think if it was longer, you'd support it? Because what you told me last time we spoke was basically don't waste your time, focus on the science.
Yes. So you have 4 years to get your paper out or your papers out, and those are the ones that will give you the job. Because my personal experience in all of the places where I interviewed as a potential faculty, not in a single place they have asked me if I had a K99. I mean, I guess they could check, but not in a single place they asked me, why didn't you apply for a K99 or why didn't you get a K99? They don't care. As long as your science is good, they will hire you for who you are and for your science. And obviously, if you have extra funding, that is a plus. That will count. In my experience, that was never a deal breaker. Maybe to get your foot on the door, that is where a K99 matters the most. But honestly, if they only want you because you have money, I mean, do you really want to work there?
And you said a lot of your colleagues who were also on the interview train, the successful ones didn't have them either, right?
Yes, because if you're busy trying to publish, a big paper, the likelihood that you will have a big paper published by the time you are able to submit a K99 is very slim. I actually got good feedback from my application, but the reason why I never got a K99 was stated in both my first submission and my resubmission was that I didn't have a first author publication. And my Nature paper came out a month after I got that letter of rejection from the NIH. So it felt unfair because I was actually asking to do exactly what I've done, which is to get trained on spatial transcriptomics to follow up on the observations that I had done. So I felt like I had a pretty compelling argument, but the money that they could have given me, they gave it to somebody else who maybe used it much better than I would ever do. So I'm not resentful. I just think that the system could be improved by a lot.
One thing I like to talk about with people on the podcast too is usually I ask the question, like I frame it as like a favorite failure. What happened in your life where you failed, but it all worked out in the end, and kind of humanize the scientist? Because a lot of people listening, maybe an undergrad student, are gonna hear you talk and be like, "Wow, this guy is so impressive! Like I could never." He's superhuman, like we started at the beginning, Superman, So I like to humanize the people a little bit. Are there any other stories that come to mind of times where it didn't work out for you?
Plenty. There are plenty stories where I just didn't get what I wanted, and that's just made me want it more and strive harder to get it. For example, when I finished my master's at Scripps, I wanted to stay in the United States to do my PhD. That was a priority for me, and I had missed the deadline for PhD applications. And I was an immigrant, so I had a visa. It was a very bad visa. It was for 6 months only, and I could not be renewed unless I left the country for a while and then came back, unless I got sponsored by a very specific visa. And anyway, so that's an immigration hurdle I won't get into, but in essence, I applied to PhD programs. I got rejected by most, and I knew I wanted it so much, but they make the applications in English. And I was 22, 23. I started actually learning English when I was maybe 18, 19.
Wow.
So when I first came to the United States, my English was poor. And maybe that I couldn't reflect that passion, those ideas I had on my application. I remember still sending some of those first emails to my English teacher back in Spain to be, hey, can you look this over? I mean, there wasn't any ChatGPT back then. I sound like an old man now, but yeah, back in my day, there wasn't any ChatGPT to do the translation for you. So maybe because of my lack of pedigree, maybe because where I came from wasn't Harvard or Stanford, it was just, the University of Barcelona, which I think is a great university, but maybe not as well known. Maybe because I couldn't express myself so much. And, Americans do have a natural talent for selling themselves. The marketing, they do it great. I just wasn't accepted. I know I wanted to go to Stanford. I applied to Scripps. I actually applied to UCSD, and I was rejected by all of those programs. I still keep some of those rejection letters somewhere. And so what I did is I stayed working on the lab that offered me the opportunity. That was Maria Diaz-Meco and Jorge Moscat at Sanford Burnham, where they say like, yeah, you want to work? You want to just work with us. And so I worked for a year without being in any program. I just worked. I was one of the best, most freeing experiences I've had as an academic, where I didn't have to submit any reports. I didn't have to care about funding. I didn't care about publishing. I only played in the lab with way more talented and hardworking people than me. And when I finally applied to the Sanford Burnham Previs graduate school program and I got accepted, I had already a massive amount of data I could just get up and running, formal PhD. I think that it wasn't a failure in retrospect because everything that happened is where you are right now. Right. And if you like where you are, that wasn't a failure.
Right. I too was rejected to a lot of graduate programs, but we don't have time to talk about all of those rejections. We're getting pretty close to the end today. I did want to end on your vision for your lab. Last we spoke, you told me about all sorts of cool projects and ideas that you had. Are you comfortable sharing some of that with the listeners today?
Yeah, absolutely. Yeah. The vision for the laboratory is that the next generation of immunotherapies for the myriad of diseases that have an immune component, which is most diseases, if you think of them, go through better understanding how immunity is deployed in tissues, how those little cells are embedded in tissues. And that's bidirectional. The immune system, the T cell, the tissue resident memory cell, needs to be adapted to work in that environment. But the tissue, the architecture of a tissue, has evolved to also accommodate that response. We don't really know how that works. We don't know as I said, what the capacity is. Is the tissue feeding the immune cell? Is the immune cell feeding the tissue? Is there a reciprocal exchange of favors? If we can understand that, then we can essentially deploy and enhance immunity in many diseases. And now we have the tools to do that. So by a combination of functional genomic screens and spatial transcriptomics and nutrient lipid uptakes in vivo, using mouse models of infection, using mouse models of cancer, and using human samples, we hopefully get a better idea of how immunity works in these tissues.
And not just the gut too, right? You're going to explore.
Yes. So the gut was an initial early foray into this world where we kind of had a good grasp on the immune side of things. The next frontier are less known tissues like the prostate or the breast, or even things that nobody think about, like the heart. There are immune cells in the heart as well. And so I have a colleague and friend at the Jolla Institute who is very interested in that. And hopefully we can get to work a bit on those less explored tissues that do have a lot of immune cells and also suffer from many diseases, as we all know.
Awesome. Well, before we come to a close here, I want to ask, is there anything else that you want to share? Anything that you'd like to impart on the listeners before we close up?
Well, I want to say they made it this far into the podcast. Thanks for listening.
Yes, thank you for listening.
Hopefully you get a better idea of who I am and what I'm doing. And I like also to say something in Spanish para todo el mundo que nos escucha desde Latinoamérica y desde otros países en las que se habla español, que no os eche atrás el hecho de que en América sean tan damn, I'm a loss for words now. It's a switching. Yeah, these are a good example. So, no os preocupéis de el habla, del idioma, si encontrar la motivación que os guste y encontrar una pregunta que os interese y que os motive a seguir. Y si tenéis cualquier duda o pregunta, que sepáis que tenéis un aliado en la Jolla Institute for Immunology y me podéis contactar en mi página web o en mi correo, y estoy más que encantado de ayudaros a venir a Estados Unidos a estudiar o daros consejos o lo que pueda ayudar, lo intentaré. So what I essentially said is that
Yo comprendo un poco.
Okay. What I essentially said is that for those who don't speak English very well or are outside of the United States, that please feel free to reach out. And they have an ally here at La Jolla Institute for Immunology, and I'll do my best to pay what some other people did to me many years ago, and I'll try to pay it forward.
And yeah, you're just starting your lab now, and you're working on building your team. So if people want to be a part of your journey and conquer the spatial world of immunology and the tissue resident memory T cell, this is a great place to do it here in La Jolla at the La Jolla Institute. So you're on social media, you're on Twitter or X, whatever you want to call it now.
Yes.
We can put that in the show notes, and people can find you on there and reach out to you and ask questions if there's questions. And thank you so much for doing this. It was fun.
Awesome. Thank you to you again. Appreciate it.